Welcome everyone again to another lecture in the nervous system, and today I will give you an introduction about the microscopic structure of nervous system. So these are our learning objectives, and let us start with the nervous system. So before talking about the nervous system, let us imagine that we are talking about an organ in our body, and the organ composed from tissue, and the tissue composed from cells. So back again to the nervous system. So the nervous system in general composed from a nervous tissue. And again, the tissue composed from cells, right? So the nervous system composed from a nervous tissue that mainly this tissue composed from cells, mainly the neurons and neuroglia cells. So you can say neurons and you can say nerve cells, alternatively. While mostly we use neuroglia or we say glia cells. That means the nervous system composed from neurons and the glia cells. And the glia cells are supporting cells to the neurons, right? In this diagram, just briefly, you see here is the PM matter, which is one of the covers that protect the brain, and here is you see the blood vessels there, the pale vessels, and here is you see the neurons. This is like a diagram that merely summarizes the components of the nervous system. Look at the neurons here, and this is the neuron, this is another neuron, and surrounded by supporting cells or glia cells. Glia cells, if you watch the previous lecture, you will understand what I mean. We will talk about that also more in details. But for now, look at the astrocytes here. Look at the microglia cells here. Look at the, I'm looking for this again, astrocytes. Look at these, also glia cells, oligodendrocytes. This is again oligodendrocytes, right? So these are glia cells, or we call supporting cells. So this is mainly, plus of course, if we want to talk about the glia cells here, we cannot ignore the ependymal cells, right? So this is like a summary of the nervous tissue. So as we mentioned that the nervous system composed from neurons and the glia cells. Let us start with the neurons. So the neurons or nerve cell, as I mentioned, these are, as you see here on the right side in this diagram, this is a neuron in which we can say it's a kind of a specialized cell forming the brain tissue, and its main function is to transmit nerve impulses, or let us say in general, before we dig deep, it transmits the what we call it message, right? So when you look to the neuron, guys, you will see that it has a couple of regions or structures. So first of all, you have the cell body, like any cell, right? And it has processes. Processes, that means something projected out from the cell body. So again, this is the cell body, and the processes can be summarized in dendrites. You see here is the dendrites, and an axon. This is the axon, right? And these, like the axon, will divided at the end into axon terminal. Anyway, back to the cell body. We'll talk about each part, but for now, this is the cell body that, let me erase this. Okay, so in the cell body, you can see there is a nucleus, and inside the nucleus, there is, apparently here we have nucleolus, right? We'll talk about both of them. And you have to know that the cell body is like the metabolic center of the neuron, right? While look at the dendrites here, processes coming out from the cell body, and if you look to the direction of arrows, here, you will see that you can figure out that the function of the dendrites is to conduct, or is to carry the impulses toward the cell body, right? Toward the cell body, while the axon, look at the direction, these arrows show the direction of nerve impulses. That means dendrites carry signals toward the, or nerve impulses toward the cell body, but the axon carries nerve impulses away from the cell body in that direction, right? So usually the neuron has one axon, right? Although, after it emerges out, sometimes there's a collateral branch coming out from the axon itself. So this is the neuron with cell body, dendrites, and axon, and I showed you the direction of nerve impulses, either toward the cell body or away from the cell body. Now, as for example, when you talk about the kidney, you would say, okay, the structural and functional unit in the kidney is the nephron, right? While in the nervous system, the neuron is, or the nerve cell is the structural unit, of the nervous system. It's highly specialized cell, as you see, to conduct impulses from one region to another. For example, the neuron with its axon and dendrites, for example, can carry an order from the your brain all the way down to your hand, to your muscle, to your abdomen, to your lower limb or upper limb or whatever. That means it's like a connection between one part of your body to another. Or on the other hand, neurons can carry signals from your muscles in the thigh or muscles in the foot up all the way to the brain and back forth, and so on and so forth. So yes, it's great to know about the function of a neuron, and also what's also great to know about that, mainly, let us say, mainly that neurons cannot divide. Neurons have no ability to divide. That means they have no ability to do or undergo mitosis. Yes, neurons lose their ability to undergo mitosis. That means they will not divide like the tissue of the skin, right? That means once the neuron destroyed or damaged, so usually cannot be replaced by a new neuron, except, let us be specific, like except in limited exceptional cases, in which this is like a field in neuroscience. If you want to dig deep, you can read more about it. But in this lecture, I can say that in limited exceptional case, if there is damage to the neuron, and it depends on how much the damage, it includes the cell body, it's close to the cell body, it's in the axon, it's at the end of the axon or close to the axon hillock. So it depends, but mainly we can say usually cannot replace once it's destroyed. And the exception, as I mentioned, in some cases, plus recently they found, yes, there is like a neural stem cells which are very limited. The division there, yes, there would be a replacement and create new neurons, but really it's very limited and slow, right? So let us then summarize it as a general: cannot neurons, once they are destroyed, cannot usually replaced. So as maybe we know from maybe your general information, that the brain and special nervous system, and the neurons that compose the main structural unit of the nervous system, it has like exceptionally high metabolic rate. That means when you say high metabolic rate, that means the neurons need highly supply or abundant supply of oxygen and glucose. What does it mean? It means in case, for example, in cardiac arrest, or there is or respiratory arrest, or whatever, there is no oxygen or glucose reach the brain. That means, or in case, for example, of the stroke, in case of cerebrovascular accident where there is no oxygen or glucose, or a blood can pass there to the brain, that means that the neurons cannot survive because they need highly abundant supply of oxygen and glucose. So in case there is no oxygen or glucose, that means the neuron cannot survive for more than few minutes without oxygen. You can say like it's vary, but the range around 4 minutes plus minus, right? So neurons cannot survive for more than a few minutes without oxygen. Yes, that was a general introduction about the nervous system, tissue, and neurons, and what does it mean when you say neurons. And now let us dig deep in the neurons that indeed vary in the shape and the structure and the processes coming out from its or their cell bodies, right? But we can confirm that there are kind of two fundamental components for the neurons. All neurons have cell body and process. You can say process or processes. That means in this case, this is multipolar neuron from the cortex, and you can see we have processes including the dendrites, right, and the axon, right? So let us make it clear. So all neurons have neuron cell body and dendrite, or one or more processes, right? That include, of course, dendrites and axon. Okay, so we agree they have cell body and processes. Let us start with the cell body. This is the cell body of the neuron, which is also called soma, especially when you write an article to be published sometime, or review, whatever, you use the soma. You can say also the cell body of a neuron, or you can say soma, right? So the cell bodies of neurons also, as I mentioned, vary widely in the size. They can, range from 5 to 140 micrometer, from 5 up to 140 micrometer. And I would like to remind you, when you say 5 to 140 micrometer in diameter, one micron is equal to 1,000th of a millimeter, right? 1 micron is equal to 1,000th of a millimeter. Anyway, so but also again, regardless the size of the cell body, if it's small or medium or large, if you look to the cell body, you will see there is a single nucleus. Here you can see the single in the purple color, right? In the purple color, there is a single nucleus, and inside it, inside the nucleus, there is nucleolus. And anyway, this is the nucleus, and the nucleus surrounded by a cytoplasm. Okay, we'll continue talk about the cell body. When you look to the cell body again, you see the nucleus, as I mentioned, nucleolus in the center, and also you will see a nucleus bodies, or you can say chromatophilic bodies. Chromatophilic bodies. What does it mean, chromatophilic bodies or nucleus bodies? We are, guys, talking about the rough endoplasmic reticulum. Rough endoplasmic reticulum. Also, we'll talk about these. Also, what we have here in the cytoplasm, guys, you have neurofibrils. Neurofibrils not shown here, but I think these like a kind of intermediate filaments to support the body, the cell body or the soma here. Also, you can see in the cell body this region, which is the axon hillock. This is the region of axon hillock, but this is the axon, right? Remember, this is the axon hillock. So if this is the cell body, where are the processes? Excellent. So coming out from the cell body, you have the dendrites. These are processes, right? Look at the dendrites that, as I mentioned, conduct or carry impulses toward the cell body. What else? Well, we have processes. We have, guys, also the axon. And the axon ends by division into axon terminals, right? And of course, sometime will be myelinated by myelin sheath. So the cell body, when you say the cell body, guys, you have to remember that it's the metabolic center of the neuron, and with its transparent nucleus here that contains a large nucleolus, as you see here in this center. So this the nucleus, the cell body, or it contains like the cytoplasm that's surrounding the nucleus, as you see, contains the usual organelles. So what we have here, we have the usual organelles that we have as in other cells, except that the cell body of the neuron, or the cytoplasm of the cell body of the neuron, lacks the centrioles. You remember the centrioles? If you remember the mitosis, you remember the centrioles and their importance in the division. And because we know that the neurons have no ability to divide, that means they are a mitotic nature. So that means there's no need for centrioles because they don't divide. Once we know this, that means the cytoplasm of the cell body of neurons have no centrioles. But you can find rough endoplasmic reticulum, or what we call it in the cell. You can find the neurofibrils to maintain the cell shape, and the neurofibrils and the cell body or rough endoplasmic reticulum are abundant in the cell body. Okay, we mentioned that already, that again, this is you see here the neurons, and I think it's a multipolar neuron, and you can see the nucleus, and inside this is the nucleus, and inside it you have the nucleolus, right? And as I mentioned earlier, that the cytoplasm here, the cytoplasm of the cell body of a neuron, contains all the urocellular organelles with the exception of centrioles, because we know there is no need, right, in this kind of cells which don't have the ability to divide. These organelles, usually supplement the other structures there, to the axon, to the dendrites, and continuously renew the membranes of the cell. Okay. Yes, we mentioned that neurofibrils are abundant in the cytoplasm of a neuron and of the cell body, of course, and they are intermediate filament or bundle of filaments that runs between the chromatophilic bodies, or what we call it, the cell bodies or rough endoplasmic reticulum. So the neurofibrils you see here is like intermediate filaments, maintain the cell shape and keep the cell like contact, not being like pulled apart when there's a case of tensile force. Excellent. So again, this is, you see the cell body, and the cell body is not just a cell or the plasma membrane. Here is not just to protect and contain um, the inside of the cell and contain and organelles there, but also it works as a receptive um, surface in which it contains like receptors to receive signals from other neurons. Okay, so um, this, this, I would say this slide is like revision for what we've been had been explained previously in the previous lecture. So, but that's fine. You know that most neurons or the neuronal cell bodies, this is the neuron, and these dendrites, and this is the axon terminal, and so forth. So the cell bodies of the neurons usually located mainly within the central nervous system. That means in the brain and the spinal cord. Excellent. So mostly the cell bodies of the neurons located mainly, when you talk, yes, brain and spinal cord, in the brain, usually in the cerebral cortex. Outside, we'll talk about it later. And also the cell bodies can aggregate like in specific regions, right, forming like a nuclei, like basal ganglia, right? For example, in the brain, in some regions here or there, there are aggregation of cell bodies of neurons. Here, they aggregated, let us say, in that region, forming something called nuclei. That means, but just you have to differentiate between what we talk about the nucleus. Listen, this is a neuron. This is the nucleus of the neuron. This is the nucleus of the neuron. But the cell body plus the cell body plus another cell body plus another cell body and another cell body, they aggregate in a region. This region called nuclei, right? Or nucleus, brain nucleus, which is like a small region where the cell bodies aggregates in one region inside the central nervous system. Can be in the brain, can be in the anywhere in the spinal cord, and we will talk about that later in the next lectures. But if the cell bodies of the neurons aggregated outside the brain and spinal cord, that means outside the central nervous system. Whereas I mean if the cell bodies aggregate in the peripheral nervous system outside the central. That means in the peripheral nervous system, we call it ganglia. The singular one is ganglion. That means aggregation of cell bodies of neurons inside the central nervous system, inside the brain and the spinal cord, we call it nuclei. But if they are aggregated in the peripheral nervous system outside, for example, this one, we call it ganglia. Here is the location, for example, of aggregation of cell bodies outside the central nervous system. Now, we finished the cell body, yes, but the cell body, as you see here, has processes, as you see. So, we have a cell body and we have the neuron process or processes, which is like processes extended out from the cell body of all neurons. So we have two types, guys, of the processes. When you see here, you see the dendrites coming out from the cell body, and also you have the axon, as I mentioned earlier. So just to keep in mind, guys, that the cell process of the neuron, as you see here, are described here using a motor neuron, which is the typical neuron that we use it to describe the different features of the neuron. But after a couple of slides or later in this lecture, we will talk about different types of neurons and how we classify them. But for now, keep in mind that we're talking about the typical neuron, which is the motor neuron, right? But the sensor neuron differs from the typical pattern. Okay, so let us agree that this is the neuron, and this is the cell body. And from cell body, we explain the structures of the cell and organelles here in cell body, and we mentioned that there are processes coming out from the cell body, including dendrites and axon. Now, let us start with the dendrites. These are the dendrites, which are like short, tapering, diffuse out from the cell body, and as I mentioned in the motor neuron, motor neurons have many dendrites clustering around the cell body. So the function of these dendrites, you see here, is they are working as a receptive sites to, communicate and carry signals from other neurons, by, of course, communicating with their dendrites or with their cell bodies or axon or whatever. It depends on the synapses there, but they are working at receptive sites and increasing by, with these branch, they are increasing the distance or area for receiving signals. That means instead of keep the cell body smooth like this, say, for example, if there are no dendrites here, so it'd be like the area will be limited and crowded. But by these branch and arbiters, that means there are many neurons with their processes can communicate at multi-dimensional pattern with this neuron. And as you see the arrows here, it shows the direction of the signal impulses in which the electrical signals by the dendrites moved or transmitted toward the cell body. But you know that the axon carry or conduct electrical signals away from the cell body anyway. So the dendrites conduct electrical signals toward the cell body. Look at this neuron. This is the cell body as you see here, and these are dendrites. Look at the dendrites here. Look at the dendrites. This is the first one. These are dendrites, and if you get focus more on dendrites, you will see there's like a small projections here. We called dendritic spines. These spines, guys, these spines are important, in which it represents like an area for close contact with other neurons, right? So the synapses synapses occur there, and they create like an area for contact with other neurons and other processes. Okay, that was about the dendrites, although there aren't much to say about it, but we would like to make the long lecture short. Anyway, that was about dendrites. Now, what about the axon? This is the axon you see comes out from the cell body, and from earlier slides that each neuron has only one axon that you see here that arises from the cone-like shape area. We call it axon hillock. This is the axon hillock. It's like cone-like cone-shaped structure, right? This is the axon hillock. So it narrows to form like a cylindrical shape, cylindrical process that relatively stays like uniform in diameter the rest of its length. And maybe you know that the axon or the length of the axon varies like between, sometimes it's absent at all, sometimes it's short, and sometimes the length can extend up to a meter, or let us say 3 to 4 feet, especially those projecting neurons from the cortex all the way down to the lower and upper limb, right? What else? Yes, this is a neuron, this is a nerve cell, and this is the axon. And the axon, sometimes we call it the nerve fiber, and many axons form like what we call it nerve fibers, right? So this is the axon and this is the nerve, we call it nerve fiber. And as I mentioned, the impulse generated an area here and in the axon at the area called axon hillock. So the impulse generated or the action potential generated there and conducted to be transmitted away from the cell body, which is, you remember that the dendrites conduct impulses toward the cell body while the axon conducts the signals away from the cell body. So if you remember when we talked about the cell, the cytoplasm of the cell body of a neuron, we mentioned that there are plenty of chromatophilic bodies, or what we call Nissel bodies, and Golgi apparatus. But indeed, in the axon and the axon hillock at the beginning here, this is the axon hillock and this is the axon. So there is no chromatophilic bodies or Nissel body or Golgi apparatus. What does it mean? Well, it means that the axon has no ability to synthesize for what we call a protein. So the protein that needed for the axon will come from the cell body. That means it will be synthesized in the cell body, then transferred to the axon, because the axon has no ability to synthesize a protein, because there is no ribosomes there, right? And you remember the rough endoplasmic reticulum and Golgi apparatus, and plus the ribosomes. All of these organelles are not exist in the axon, so the axon lack ribosomes and all organelles I mentioned involved in the protein synthesis. So the axon has no ability to synthesize protein by itself. So they must receive their, let us say the axons must receive their protein from the cell body, as I mentioned. But on the other hand, yes, the axon has no ribosomes, chromatophilic bodies, or Golgi apparatus, has no ability to synthesize a protein, but it has neurofilaments, actin microfilaments, microtubules, and they are evident in the axon because the axon need to be supported and structurally, I mean. So these filaments create a kind of structural support and strength. These we call them cytoskeletal element. Also, they are help. These cytoskeletal elements held in the transport, not just support the axon, the structural support, but also it helps in the axonal transport, for transport of substances to and from the cell body. Yes, as I mentioned, every neuron has mainly one axon, and well, it does happen that a kind of axonal collaterals, like a branch from the axon, can come out from the axon, and we call it axon collateral, right? And sometimes there are more than what we call axon collaterals, right? And they extend from the axon at almost right angle. We continue. We started from the axonal hillock here, and then this is the axon, and this is the axon collateral. Then you go all the way until you reach the end of the axon. Then the axon here terminates by dividing into what we call it axon terminal. Axon terminal. Look at here at the synaptic end pulp. These pulps contain neurotransmitters. Right, to be secreted and released once the action potential reached, right? So the axon ends in what we call axon terminal, and here's in the pulp for what we call it synaptic end pulp. As I mentioned, it contains neurotransmitters for example, like glutamate. But there is, if you say, of course, this is the neuron, and there is another neuron here. This is the dendrite, cell body, and axon, and these are the axon terminals, right? So you have to keep in mind that there is a gap here between the axon terminal of this neuron and the axon terminal of the other. Or let us say, if there is axonal terminal here, and the cell body is here, and dendrites is here, right? So there is a gap here. This gap, slit gap, which is like a small space for neurotransmitters to be released there, right? Anyway, again, I would like to iterate that the nerve impulse arises at this region for what we the cone shape, what we call axon hillock, and sometimes it's called spike initiation zone. Spike initiation zone. Okay. So generated the nerve impulse here at the axon hillock, transmitted through the axon all the way until it reaches the axon terminal as you follow the arrows, and then a neurotransmitter here is like released into the space, to the extracellular space, to trigger the next neuron, and so on and so forth. These neurotransmitters, it depends, they can be excitatory or inhibitory, like for example glutamate when excited, or GABA when it inhibits the next neuron. Or it can be not just a neuron, can be targeted organs, can be the skin, can be the muscle, or whatever, right? So finally, when we talk about the axon, I would like to remind you that not just the length of the axon varies from a neuron to another, but also the diameter of the axon itself. So while they are vary in the diameter, that means we have to expect that the larger diameter, the axon with the larger diameter, conducts impulses for longer distance than those with the smaller diameters. That means the axons with larger diameters conduct impulses for a longer distance compared to smaller diameter. And if you would like to dig deep about the effect of the characteristics of the axons, whether like the diameter, the effect of myelination, that means the axon myelinated or not, the role of node of transverse on the effect on kinetics of action potential, speed, for example, of action potential, you can find any neuroscience textbook, and I highly recommend this textbook for John Dowling. It's a very nice textbook that titled by Neurons Network, an introduction to neuroscience. This is the first edition, and I think recently the second edition released. Okay. Getting an idea about the different structures of the neuron, let us identify how neurons be classified. So, well, indeed, basically the neurons can be classified into either structurally or functionally. That means depending on structure, or it depends on its function. So let us start first with the structural classification of neurons. How do we classify the neurons according to their structures? Well, indeed, we classify neurons according to their structures according to the number of neurites. What does it mean when you say neurites? Well, you are talking about the axons and dendrites. The number of axons and dendrites, both we call them neurites. So depending on the number of neurites, we classify structurally the neurons into three types: multipolar neuron, bipolar neuron, and unipolar neuron. If you look at these terms, you see multi, that means many, bi, that means two, uni, that means one, right? So let me show you here the structural classification of neurons. Here is an example of multiple or multipolar neurons. Here's an example of bipolar neuron, and the last one on the right that shows the unipolar neuron. Okay, so the multipolar neuron, if you look at it here, look at the cell body. When you look at the cell body, there are many processes, not one, not two processes. No, there are many processes coming out from the cell body, including, look at many dendrites plus the axon. This is the multipolar from its name, multi. While what about the bipolar neuron? From its name, it shows that there are just two processes coming out from the cell body, in which they extended like in the opposite direction, as you see here. One would be the dendrite, and one would be the axon, right? So two processes extended from the cell, one fused dendrite, then this fused dendrite, it gives like a many branch like a tree. Anyway, on the other pool, you have the axon. This is why it's called bipolar neuron. But the unipolar neuron, this is the cell body. From the cell body, there's just one process extend out from the cell body and forms like T-shape. Look, it's like T, right? So this just one process coming out from the cell body, then it divides, as you see here, forming peripheral and central processes. This is the peripheral process, right? And this is the central process. You know what I mean? So because these are so we can we'll talk about each one of them, but let me here add one thing, that the peripheral process, look at the direction of impulses. That means the peripheral process, this part conducts impulses toward the cell body. That means it's a dendrite. So we will say, okay, this is a dendrite. While this central part, you see the direction of the impulses, signal impulses, away from the cell body. So we can conclude, yes, this is the axon, right? So the peripheral process would be the dendrite, while the central one would be, we assume that it's the axon. So let me give you a little bit of detail about the each one. So the multipolar neuron, this is here you see one example of multipolar neuron. Look at many processes coming out from the cell body, including dendrites and one axon. And this type usually, the multipolar neuron represents like most of the neurons in the body, like 99% almost, right? So usually in the brain and spinal cord. Well, we cannot ignore that some multiple neurons, well, a few of them, I would say, they lack an axon, right? They don't have an axon. For example, an example of multipolar neurons is the pyramidal cells in the cerebral cortex, or Purkinje cell, as you see. Look at them. So this is a cell body. There are many processing coming out, and on the right side with the pyramidal cell. It's called pyramidal cell because the cell body is like a pyramid, right? Okay. Now the second one. So in the multipolar, we got like we explained where is it, and we show like two examples on the multipolar neuron. Now, what about the bipolar neuron? As I mentioned, this is an example of bipolar neuron, the cell body in the center, and there are two processes coming out from in the opposite direction from the cell body, as I mentioned. So this kind of cells, like rare, we consider like rare type of neuron that usually found in the special sensory organ and usually function as a receptor. What does mean? Well, it means that because it functions as a receptor and in a special sensory organ, usually you will find it in the retina of the eye, and the retina of the eye, which is like what we call bipolar neuron of retina of eye, right? This is number one. So it receives like signals from here, then directed toward the brain, and also in the inner ear. Plus, also, it's not just in the retina, like for what we call it bipolar neuron of retina, right? But also in the epithelium of olfactory mucosa, right? Olfactory mucosa, those like sensing the smell and particles there, then the signals will be transmitted toward the axon and then ultimately to the brain, right? So it exists in they function, the bipolar like a rare neuron, function as a receptor, usually in the special sensory organ like retina and those in the olfactory mucosa. Now, lastly, the unipolar neuron. This is the unipolar neuron. Look at the cell body. There is just one short single process coming out from the cell body. It forms like a T-shape, as I mentioned earlier, and usually found if you later, after a couple of lectures, we will get an idea about the anatomy of the spinal cord, and this is a cross-section of the spinal cord that shows, like here there is a bulge here called dorsal root ganglion. Dorsal root ganglion. So ganglion, as I mentioned earlier in this lecture, aggregation of cell bodies outside the central nervous system, I mean in the peripheral nervous system, we call it ganglion, right? And in this case, this cell body is located here, right? And from there is one, you see here, look at the blue one, here is the cell body, then it gives like central one and peripheral process, central process and peripheral process. So the peripheral one is like dendrites that carry signals from the skin, from muscle, whatever. Look at the direction of the impulse to the cell body. From there, the sensation will go to the dorsal horn in the spinal cord by this axon or the central part. Usually, the function of unipolar is a sensory neuron. That's like extra. Anyway, now, as we mentioned, motor, sensory, whatever, so we jumped, let us jump to the functional classification of the neurons. Earlier, we did classify the neurons structurally into multipolar, bipolar, and unipolar. Now, what about the functional classification of neurons? Indeed, we classify neurons into sensory, motor, and interneurons. Sensory, we call them afferent, from A, right? Motor neurons, they carry signals from the brain and central nervous system to the periphery. We call it efferent because they exit the central nervous system. And there is in between what we call it the interneurons. We call it interneurons. So let us start with the sensory neurons. Again, the functional classification of neurons will relatively depends on the direction in which the nerve impulses travel relatively to the central nervous system, brain and spinal cord. So the sensory neurons, we call them the afferent neurons. Afferent, that means toward the. That means the sensory neurons, we call them the afferent neurons because they transmit the signals and impulses toward the central nervous system from the sensory receptors in the peripheral nervous system or from different parts of your body. For example, here in the skin, there is a receptors here for pain, temperature, touch, pressure, whatever. So the signals or signal impulses will be transmitted toward the central nervous system, toward the spinal cord and the brain. And you see here is the blue color that represents the unipolar neuron. You remember the unipolar neuron in which the cell body just one process coming out from there, and it's located outside the central nervous system. Usually, we said its function is or this kind of neurons work as or function as sensory receptors, right? Or sensory neurons carry signals toward the central nervous system. So this is the unipolar neuron, which is the cell body located in the ganglion. Here is the peripheral part that works as dendrite, and this is the central part as an axon that carries the signals to the dorsal root ganglion in the spinal cord. That means in the central nervous system. Yeah, we explained that previously. And now what about the motor neuron? Well, indeed, this red color indicates or represents the one example of motor neuron. We call it efferent neurons because this kind of neurons transmits the signals from the central nervous system to the periphery. That means this kind of neurons carry impulses away from the central nervous system toward the periphery, to the muscles, to the gland for secretion, and etc. Usually, the motor neurons can be divided into upper motor neuron that, for example, that comes out from the brain. So this is the cell body, for example. This is the upper motor neuron, and here is the lower motor neuron. Usually the upper motor neuron located in the central nervous system, and the lower motor neuron located in the peripheral nervous system, right? We will get more details like in the next lectures. So usually the motor neuron, as you see here, the red one, the motor neuron, usually multipolar neuron, which is this is the structural classification, but anyway, and their cell bodies, as you see here, located in the central nervous system, in the brain or spinal cord, in this case in the spinal cord, except the autonomic nervous system, where it's located like out the central nervous system. So the motor neurons, as you see in this case, like form a junction with the effector cells and signaling, for example, muscle to contract or the gland to secrete their secretion. Now, in between, there is functionally, we not just have sensory and motor neuron, we have in between the interneurons, or we call it association neurons, right? In this case, look at it here in the green color. So almost I would say interneurons are multipolar neuron, and they are these kind of neurons, like between the motor and sensory, it's like the ring or the connection in between. So they creating a kind of a complex network there in the central nervous system. And what's interestingly to say that the interneurons, guys, show like great diversity in the size and the branching pattern of their processes. Let us move to another type of cells in the brain, for what we call it the neurocells. glia cells, or what we call it supporting cells. Let me remind you that, guys, before a couple of slides, we talked, or at the beginning of the lecture, we talked about the nervous tissue, and we said that the nervous tissue is as a tissue formed from cells. Mainly you have neurons and you have glia cells. So now we finished the neurons and different structure and characteristic and the classification of the neurons. Now let us shift to the glia cells, or for what we call it neuroglia, or sometimes we call it supporting cells. We call it supporting cells because they support the neurons, and each one has distinct function. So one of the there are a couple of characteristics for the neuroglia cells in general. One of them, that they are not electrically excitable, that means they are not like neurons. And although they are not electrically excitable, but they form like about half the volume of the nervous system. Look at them here, 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 here as well. So this is the second thing. The third thing, which is that neuroglia cells can multiply and divide. What does it mean? You remember that the neurons cannot divide, right? Neurons have no ability to divide, but glia cells, indeed, they have the ability to multiply and divide. And from here comes the problem of the brain tumor. So brain tumors, brain cancer, brain tumors come from the glia cells, not from neurons, because glia cells can divide and multiply, and from there, tumors can arise there. Anyway, so back again to neuroglia cells. They are, we have 6 kinds in total. 4 you see here in this figure is the cross-section of the nervous system in this figure, and we don't have the peripheral nervous system now, but anyway, we have 4 types or kinds of glia cells in the central nervous system, and we have just 2 in the peripheral nervous system. So the neuroglia cells in the central nervous system that you see here are the astrocytes. You see, this is the astrocyte. There are 2 types of astrocyte, one called fibrous astrocyte and protoplasmic astrocyte. We will talk about them. And we have a kind of like a spider-like shape, which is the microglia cells. It's like a spider shape. And we have ependymal cells that you see here that usually lines the cavities in the brain and spinal cord. And we have oligodendrocyte. These are oligodendrocyte that myelinate the axons in the central nervous system, and we'll talk about it as well. While in the peripheral nervous system, guys, we have just 2 types, Schwann cells instead of oligodendrocyte, and we have satellite cells. So these 2 types of neuroglia are found in the peripheral nervous system. Okay, let me just also give you a brief idea about the neuroglia cell in general, as they are supporting cells and they have a unique specific function, but in general they provide a kind of supportive scaffolding for neurons in the nervous system. In addition, if you look at them, while they are supporting, but also they are create a kind of insulation for the neurons, especially in the area where there is no electrical activities, in which this when you insulate the neurons, that means you keep the electrical activities there, and from like protecting them from adjacent neurons not to interfere with each other, right? It's a kind of insulation, right? If you look to the neuroglia cells in general, you will see that they have like processes. Look at the processes here. So yes, they similar to the idea of the processes of the neurons, with like centrally cell body. But what distinguished it from neurons that usually they have smaller size compared to the neurons, right? They are smaller in size, right? Plus, so they are supporting the neurons. They have many functions. They can divide and multiply. Um, they have branches, but they are smaller in size, but larger in number. So they outnumber the neurons like in a ratio of 10 to 1. That means you have 10 glia cells equal to like in number, when you have 10 glia cells compared to one neuron there, right? So the ratio is 10 to 1, glia to a neuron, right? That means they make up to the half of the mass of the brain in general, right? And they have the ability to divide throughout the one's lifetime. So that was in general. Again, let me show you the neuroglia in the central nervous system before start talking about each one. So you have the astrocytes in the purple or blue color, let us say, the protoplasmic ones, and the fibrous ones. So these are astrocytes. You have the microglia cells, which is like spider in shape, spider-like cells, ependymal cells again, and oligodendrocyte. The first one, which is the astrocyte. Astrocyte from its name, astro it means like a star, cyte it means cells. So that means this is a green one. I will use the green color. So this is the astrocyte in this figure you see, in which it's like a star in shape, multiple processes, and it's abundant in the central nervous system. They are supporting because they have microfilaments that gives microfilaments that give the astrocyte the ability to support the surrounding neurons. This is number one. And also look at the feet of the astrocytes processes. These feet, guys, these feet attached to neurons and to the surrounding, as you see here, to the neuron and to the surrounding blood vessels. I will show you after one or two slides. And by this way, it the astrocyte considered as one of the cellular components of the blood-brain barrier that regulates the transport of substance from the blood to the brain and vice versa. Somebody can say, what's the blood-brain barrier? Blood-brain barrier, it's the anatomical and physiological selective protective barrier in the brain. If you look inside the brain here, you will see like capillaries, blood vessels. These blood vessels, found by endothelial cells. These endothelial cells connected to each other by tight junction. So when the blood, inside these blood vessels, there is a blood. So the blood, the brain doesn't allow for the different substances by different concentrations to pass from the blood to the brain and vice versa, from the brain to the blood, because the brain is very sensitive organ. That means the concentrations of the different materials, different ions, different chemicals in the blood, it's not suitable to be in the brain, right? So the concentrations of those ions and materials and stuff like that in the brain different than that in the blood. That means the blood vessels in the brain need to be highly selective and not allow for any substance with different concentration to pass from the blood to the brain. This endothelial cells forming the blood capillaries here, or line the blood capillaries with the tight junction there, plus look at the astrocyte, the feet of the astrocytes around the blood vessels with pericyte here and the neurons here. All of these are cellular components forming the blood-brain barrier. That means endothelial cells in the capillary, pericytes, astrocytes, neurons, all of these form the blood-brain barrier. So, what I want to say that, yes, the astrocytes participate by their feet to be like part of blood-brain barrier that physically and chemically regulates the passage of substances from the blood to the brain. This is number one. Also, it regulates the diameter of the blood vessels. For example, they have feet connected to neurons. When there is increase in the electrical activities in the neurons, that means the brain and neurons need more blood and more energy, more oxygen, more glucose. So by their other feet connected to the blood vessels here, which is part from the blood-brain barrier, it simulates in a way or another the blood vessels to dilate and increase in diameter to increase the blood flow and oxygen to the brain to accommodate the increase in the neuronal activity. By this way, it has, our astrocytes have a role in regulation of the cerebral blood flow, right, to the brain. Plus, when there is like increase in the excitation of the neurons, you know that the extracellular potassium increased and the neurotransmitter for what we call the glutamate also increased in the extracellular space here. So the potassium ions and the glutamate will be buffered. That means reabsorbed by the astrocytes. By this way, it regulates or controls the chemical environment of the brain in a way where we call it buffering system. So it buffers the extracellular space of the brain by reabsorbing and regulating different ions and substances, including the potassium and glutamate. Okay, already we explained that. Furthermore, even like earlier during the embryonic and fetal development, the astrocytes involved with the synapse formation. How is that? How is the astrocyte involved with synapse formation? Indeed, by during the developing neural tissue, by producing molecules that necessary for neural growth, or what we called brain-derived trophic factor. Brain-derived trophic factor, and by this way, it increases like and enhances the synapse formation during the developing of neural tissue. That was about the astrocytes. I think maybe we forgot to talk about the protoplasmic astrocytes and fibrous ones. Well, mainly the protoplasmic astrocytes is like a star in shape. The processes are branched and short, as you see here, short and branched, and usually located in the gray matter. Say, for example, the cerebral cortex, right? This is the protoplasmic. While it's not exclusively, it can be in the white matter, but mainly in the gray matter. Now, the fibrous astrocytes usually they have long and unbranched, relatively, processes compared to the protoplasmic, and usually it exists in the white matter. Now that was about the astrocytes, but what about the microglia? Yes, this is the what we call it. Let me use. Okay, I will say. Okay, this is the microglia, which is like spider-like shape. Indeed, the microglia are elongated phagocytes, so their function mainly, or it works as a phagocytes, in which let us call it the macrophages of the central nervous system. You know, we have macrophages in our blood, and you have macrophages in the brain. The macrophages of the central nervous system, brain and spinal cord, indeed, is the microglia. So yes, the main feature of it, other than it's like a spider-like shape, elongated shape, but also it's the smallest glia cells, and it's the least abundant type. Why it's the least abundant type? Because it passes or derived from the blood cells and migrate to the central nervous system during the embryonic period, right? During the embryonic and fetal development. That's why it's limited. Their number, or they are limited in number, right? Anyway, the main function, they work as, or they function as phagocytes, in which they remove the cellular debris there, for example, after neuronal damage or for in a way or another. So they remove the cellular debris and phagocytize the microbes and damage nervous tissue. So they are really active all the way, and especially during neuronal damage. So again, this is I think this is gold stain showing a microglia, and again I would like to iterate that they are derived because they are the least abundant type of neuroglia cells, because they are derived from blood cells and they migrate to the central nervous system during the embryonic and fetal development. But after that, no way to get in the brain, right? Okay, what we have other than the astrocytes and the microglia, we have ependymal cells. Look at this row of cells. These like cuboidal to columnar shape arranged in a single layer, and of course they have a microvilli, or let us say cilia here, in which these cilia, you know, here is like we have a CSF, cerebrospinal fluid. There is a cavity here. So they, what you look at it here. This is the central canal in the spinal cord. There is a canal in the central of the spinal cord. If you take a cross-section of spinal cord, you will see there is a small hole here. This hole is the central canal. Look at the central, around the central canal, there is a cuboidal to columnar cells arranged in a single layer. These are ependymal cells. These are ependymal cells with cilia there. So ependymal cells line the ventricles. When you say ventricles and canals, like the cavity in the brain and spinal cord filled with cerebrospinal fluid, right? So by their cilia, the CSF moved. It's not the only way by which CSF moved, but one of the function of the cilia of ependymal cells is to move the CSF inside the ventricles and spaces or cavities in the brain. Again, not that much to say about it. These ependymal cells arranged in this way, and of course create a kind of a barrier between the CSF, cerebrospinal fluid, that's in the cavity, and the brain, forming like what do we call it, CSF brain barrier. This like permeable barrier between the CSF that's located in the cavities there and the cells of the central nervous system. The last type of glia cells or neuroglia cells is the in the central nervous system is the oligodendrocyte. Oligodendrocytes, look at the oligodendrocytes here. Let me use this pen. Okay, look at the oligodendrocytes. It's the main function of the oligodendrocytes. It's exclusively in the central nervous system, so it forms myelin sheath around the axons in the central nervous system. Listen, I'm saying around axons because each oligodendrocyte has the ability to myelin more than one axon, right? And the oligodendrocytes located exclusively in the central nervous system, not in peripheral, right? Which is important. They are fewer like branch if you compare it to astrocyte. Here you can see it's like fewer branch. Usually their processes, as you see here, wrapping around tightly around the axon, especially the thicker in the thicker neurons in the central nervous system, and they produce like a myelin sheath insulating the axons and covering it, right? Now, that was about the central nervous system, but what about the peripheral nervous system? As I mentioned earlier, we have two types of cells, Schwann cells and satellite cells. Indeed, these cells are similar in type, let us say, but they are different mainly in the location. Yes, they are both in the peripheral nervous system, right? This is the Schwann cell you see here, and here is the satellite cell. You remember this kind of neuron, unipolar neuron, that's located in the dorsal root ganglion? Do you remember that? Anyway, so the Schwann cells, guys, you see here, I'm talking about the Schwann cells. Each Schwann cell myelinates one axon just in peripheral nervous system. You remember the oligodendrocyte in the central nervous system? Each oligodendrocyte in the central nervous system myelinates more than one axon, right? But in the peripheral nervous system, look at the axon here. This is unipolar cell, just to know. This is a unipolar cell. Anyway, this is a cell body, and this is the single process with peripheral and central processes. Anyway, so this one cell forming a myelin sheath around the nerve fiber. You see? This is number one. And also, what's the most interestingly about the Schwann cells, they are participate in axon regeneration. For example, during damage to the nerve or the peripheral, one of the peripheral nerves, so Schwann cells creates a kind of a wrap or a fold that facilitates the regeneration and the growth of the axon again for regeneration, which is like way let us say simpler than or easier than the regeneration in the central nervous system. So the regeneration in the peripheral is like way easier than that in the central nervous system. Anyway, this is about the Schwann cell. That means it's around the axons. But what about the satellite cell? You remember the unipolar cell in the dorsal root ganglion? This is the cell body. The cell body, listen, the cell body, not the axon. The cell body of this kind of cell surrounded by satellite cell, right? So satellite cells located in the peripheral nervous system ganglia, in the peripheral nervous system, ganglia surround the cell bodies of the neuron there. And by this way, it believes that the satellite cells regulate the exchange of material between the neuronal cell body and the interstitial fluid there, right? Although the function of satellite cell are largely unknown. We talked, guys, about the neurons and the different structures of the neuron itself, and we mentioned a couple of things about the axon, and we mentioned that some axons are myelinated and covered by myelin sheath. And also, if you remember, we talked about the glia cells in the central nervous system and in the peripheral nervous system. And we mentioned that we have Schwann cells in the peripheral nervous system that's responsible for creating a kind of myelin sheath around the nerve fibers in peripheral nervous system again, while the oligodendrocyte is responsible, responsible for making myelin sheath around nerve fibers in the central nervous system. That means, yes, both are glia cells, but one works in the peripheral nervous system, I mean Schwann cells, and one in the central nervous system, and I mean here the oligodendrocyte. So the myelin sheath in the peripheral nervous system, like it's produced by the Schwann cells, as you see here up in the figure. So what you see here is a Schwann cell in the peripheral nervous system. I don't have one for oligodendrocyte, but after a couple of slides we'll talk about it. And you know, the myelin sheath ultimately will surround the axon of most neurons, as you see here. So one thing also I would like to remind you that in the central nervous system, yes, the oligodendrocyte is responsible for myelinating the axons, and each oligodendrocyte can myelinate several axons. But in the peripheral nervous system, the Schwann cell you see here, one Schwann cell can myelinate just one single axon, as you see up in the figure. So one more thing to mention here, that yes, the myelin sheath is not continuous. If say this is the axon, so the myelin sheath is not continuous around the axon. That means you have gaps here, right? These nodes, we call it nodes of Ranvier. Nodes of Ranvier. So the nodes of Ranvier are important for conduction of impulses. Okay. So although it's written here that the myelin sheath is like one of the function of the myelin sheath is to insulate the axon and reserve and support the conduction of action potential, but indeed, yes, it's written here the myelin sheath insulates the axon and increases the speed of nerve impulses conduction. Well, indeed, I would like to delete this because recently it has I would say it turns out that it turns out not to be an issue. That means it's not really important to be myelinated or unmyelinated as a factor to increase the speed of nerve impulse. Well, indeed, what increases in reality, what increases the speed of nerve impulses is the distance between nodes of Ranvier. The nodes of Ranvier. The distance, usually in mammals and humans and so forth, it's about 1 millimeter, but sometimes, can be more or less. So the shorter ones are the better, you will get better like speed. So it's not really important if it's myelinated or not, although there are many textbooks talking about the myelin sheath as as a main factor to increase the speed of nerve impulses. Yes, it helps, but it's not really significant. Anyway, there is a textbook if you need to dig more. John Dowling, he has a textbook published, I think the second copy, second edition, yes, published recently. Anyway, so guys, you see here is the multipolar neuron here, and look at the axon, and the axon is like myelinated, and look at the myelin here, look at the Schwann cell, because it's in the peripheral nervous system. So you see here is the Schwann cells, right? This one, look at the nucleus of the Schwann cell here, and this is the cytoplasm of the Schwann cell, and the outer membrane of a Schwann cell here, for what we call it here, what we call it neurolimma. This is the neurolimma, the outer cytoplasmic membrane of Schwann cell, right? So you have the outer membrane, or what we call it neurolimma, the outer cytoplasmic membrane, and you have the nucleus here, which is like oval or flat in shape. And the Schwann cell itself, you know, the plasma membrane of a Schwann cell, it creates like a sheaths around the axon itself that you see here. These folds is the myelin sheath, Again, here's the Schwann cell, myelinated the axon. This is again, let me erase it and show you here. This is the Schwann cell in general, myelinates the axon. So this is a myelinated axon. And this is the nucleus of a Schwann cell. This is the cytoplasm. This is the cytoplasm of Schwann cell. And this outer layer is the cytoplasmic membrane of a Schwann cell. We call it neurolimma, right? But what about the plasma, the plasma membrane itself? The plasma membrane, guys, here wrapped around the axon, create for what we call it myelin sheath. This is the myelin sheath, right? This is the myelin sheath. If you remember here, look at it here. This is the Schwann cell. This is the nucleus. This is the cytoplasm of Schwann cell. Then, and this is the outer layer, neurolimma, right? But look at it here in the how the process of myelination occurs. Now look at the plasma membrane here, in which it started to wrap and fold around internally around the axon. Then it creates couple of layers. This is a plasma membrane of Schwann cell. Then the plasma membrane also fold all the way to create a myelin sheath. The myelin sheath. Okay, I think it's clear now. We explained that, and we mentioned that Schwann cells along the myelinated axon don't touch each other, so they create a kind, or they leave a gap in between for what we call it nodes of, these are the nodes of Ranvier, that occurs at regular interval about 1 millimeter apart, right? So this distance about 1 millimeter, which is important for the conduction of the impulse. So in case of, you see here the myelinated axon, guys, so the nerve impulse, you know, it moves in that way, don't travel along the myelin covered region. So instead it jumps, the action potential jumps like from node to node, right? Jumps instead jumps from the membrane of one node of Rabia to the next one, that increasing impulse conduction. So the distance between nodes of Rabia is the most important thing in the speed, right? Again, here's like written that myelin increase the speed of transmission of nerve impulses, but I explained what's been turned out recently or found recently. So anyway, in general, the myelinated axon transmit nerve impulses rapidly up to 150 meters per second, while the unmyelinated like very slow, in which the nerve impulse transmitted like about in a range about half a meter to 10 meters per second, very slow. Okay, now we talked about the I gave you an idea about the myelination, especially in the peripheral nervous system made by Schwann cell and the structures of the Schwann cell, how the process occurred, and but you know what? We have to mention that not all axons are myelinated. Yes, we have yes, we explained the myelinated axons. I'm talking about now the peripheral nervous system, right? So in peripheral nervous system, you know whose myelinated is the Schwann cell. So when you say myelinated axon in the peripheral nervous system, you are referring to the axon who's covered by Schwann cell, right? And the plasma membrane of the Schwann cell, as you see here, creates kind of or wrapping around or wrapping a coat of myelin sheath. That means the plasma membrane wrapped like making layers around the axon, as you see here. So this is the myelin sheath, right? So this is myelinated axon. We call it myelinated axon. Um, some people call it myelinated axon with neurolemma, with neurolemma, right? Example, the peripheral nerve. Okay. Now, some still, we talk about the peripheral nervous system. Some are unmyelinated. Yes, they are unmyelinated, but with what we call it with a neurolemma. Yes, unmyelinated. How's unmyelinated? We know myelinated with a neurolemma in which there is one cell, and and the plasma membrane of Schwann cell itself like wrapping around the axon, creating myelin sheath. But what about the unmyelinated? It's unmyelinated, but again, it's encased by Schwann cell. That means this is a Schwann cell. You see, this is the nucleus of Schwann cell, and this is the cytoplasm of Schwann cell. But listen, yes, the Schwann cell is here and around, and but the plasma membrane of Schwann cell doesn't wrap around the axons and creating a kind of a myelin sheath, right? No, it just encircling the Schwann cell itself, encircling the axon, but just there is a neurolemma here, Schwann cell neurolemma, but there is no myelin sheath around each one, right? So we consider it like unmyelinated. Okay. Another type I would say I would add here the what would you call it, naked fibers. Naked fibers, they are unmyelinated without even Schwann cells. Where is that? You remember the neuron, and this is the axon. You know the axon terminals. So here is the axon terminal. There is no myelin sheath at all, no Schwann cell at all. It's like this, the axon terminal, right? This is, we consider it naked fibers. Okay. Now, here is like another review for unmyelinated axon. This is electron micrograph of unmyelinated axon. Look at the outside. Here is the Schwann cell, the green one. The green one is the Schwann cell, right? And this is the cytoplasm of it. And outside you have the neurolimma, which is the what we call it the cytoplasmic membrane of the Schwann cell outside. Yes, and look at the plasma membrane. So these are axons, right? In the red color, these are axons, right? But look, there is no myelin sheath around the axons. Just there is a cytoplasm, there's a Schwann cell with a neurolimma, and there is a cytoplasm, but there is no myelin sheath around the axons. So we consider them like this, or this like what we call unmyelinated axon. Usually exists in the sympathetic, or we call it autonomic nervous system, and in some sensory fibers and nerve terminals. Now that was about the peripheral nervous system, but what about the central nervous system? Yes, we mentioned that the oligodendrocyte, which is a type of glia cells, found in the central nervous system in the brain and spinal cord, and these kind of cells, oligodendrocytes, are responsible for making myelin sheath around nerve fibers in the brain and spinal cord. I mean, in the central nervous system. Look at the oligodendrocyte, small in size, few processes, but each oligodendrocyte can myelinate more than one axon, as you see here, by its processes. This is number one. And by its like processes create, like, look at their fees, so it encircles the fibers. There is no Schwann cells in the central nervous system. Now I would like to mention one thing before jumping to the clinical correlation. I would like to talk briefly about the synapses between neurons. You see here is a multipolar neuron, like with synapses from different locations. So one of the form of synapses, like between the axon and the dendrite, in which we call it axodendritic synapse. Axodendritic synapse. Look at the axon. Here's the axon terminal, and here is the dendrites. So the synapse between the axon and dendrites, we call it axodendritic synapse. But also the axon can be the axon can synapse with the cell body. We call it axosomatic synapse between the axon and cell body of the next neuron. Also, the axon of a neuron can synapse with the axon of the other neuron for what we call it axonic synapse. On the other hand, in which it's not shown in this figure, that the dendrites of one neuron can also make a synapse with the dendrites of the, say this is the axon, right, with the dendrites of one neuron with the dendrites of the other one, what we call dendritic synapse. So yes, we talked about the myelination, and there is a well-known disease, which is autoimmune disease, what we call it multiple sclerosis, MS. Look at the healthy nerve fiber with the myelin sheath, and on the other hand, look at the affected nerve fiber in which the myelin sheath destructed and damage. In this case, we call in the central nervous system, in the brain and the spinal cord, if there is a damage to the myelin sheath around nerve fibers, we call it multiple sclerosis, which is body's immune system. T cells attack the myelin sheath and attacks the myelin sheath that protects the nerve fibers. Can be, either partially or completely strip the myelin off the fibers, leaving the nerves or nerve fibers unprotected and uninsulated. This, of course, affects the passage of the message from the brain to the other parts of your body, and so the message can be like disrupted or delayed, and sometime the message to the brain received there may be like misinterpreted, right? Can be detected, of course, using the MRI. As we talk about nerve fibers, let me remind you that the axons created in the central nervous system create a nerve or a bundle of axons here inside the central nervous system, in the brain and the spinal cord. This creates the tracts. We will talk about it after a couple of lectures. So the nerve bundles or nerve fibers in the central nervous system, they form something called tracts. But the bundles of axons or neurons in the peripheral nervous system creates what we call, or they form nerves. That means in the peripheral nervous system, the nerves formed by a bundle of axons. So in the peripheral nervous, we call them nerves. But the bundle of axons in the central nervous system, the brain and spinal cord, they create like tracts. If you take a cross section through the spinal cord, for example, you will find like tracts here, like bundle of axons, like for example anterior or lateral corticospinal tract, and so forth. We got an idea about the nerve fibers, or what we call it the axons, and we mentioned that some of them are covered by myelin sheath, as you see here on the right side in this figure. But yes, this is the axon, and the some of axons like covered by myelin sheath. Then what? Do you think there are other covers which protecting the axons and creating like bundles and ultimately a nerve? Yes, indeed. Look at the axon covered by myelin sheath, then each axon with myelin sheath covered by delicate loose connective tissue, for what we call it endoneurium. And look at them here in this bundle. So each axon with myelin sheath covered by endoneurium and in all axons. Okay, now all these axons with myelin sheath and their covers, endoneurium there, all they are covered by perineurium, which is a dense connective tissue, surrounds the fascicle or the bundle. So this is a perineurium, and this is perineurium, and this is another perineurium, and this is a perineurium. And all these fascicles, this one, all they were surrounded and covered by and wrapped by connective tissue, dense connective tissue, for what we call it epineurium. This one that forms, of course, that gives you the ultimate shape of the nerve, right? So again, you have the axon covered by myelin sheath and the axon with myelin sheath covered by endoneurium. All they aggregated like in a bundles, as you see here. So the bundle covered by perineurium, all bundles aggregated with vessels and so forth covered by epineurium outside. Okay, we mentioned that, and let me show you this histology sections. Here is again, you see the, for example, the nerve, the axons with myelin sheath, and outside you have the the endoneurium. Now, such these like endoneurium here will be ultimately form like a fascicle or a bundle that's covered by perineurium, and also this bundle or fascicle form covered by perineurium. Now these bundles all together here, another bundle here, and so forth, all they will be covered by epineurium. Very simple. Look at it here also in this figure on the right side. You have the axon here covered by myelin sheath, white color, and look at it here. This is the axon and this is the myelin sheath, and this is the endoneurium. This is the endoneurium for each axon with myelin sheath. And all of these axons with myelin sheath and endoneuriums here, all they form like a fascicle, right? This bundle or fascicle covered by, for what we call it, perineurium. This bundle and this bundle and all together, guys, of course they will be covered by epineurium, right? Look at the longitudinal section here, look at the axon here, very clear, and the myelin sheath here, right? Is like a myelin sheath, and here is the node of Ranvier, right? So again, here's the myelin sheath and the node of Ranvier, and this is the axon myelin sheath and covered by endoneurium, you see. Okay, now finally we have the nerve ganglia, and we mentioned earlier that the aggregation of cell bodies of neurons outside the brain and spinal cord, that means aggregation of cell bodies of a neuron outside the central nervous system, we call them ganglion, and more than one ganglion we call it ganglia, right? So let us talk about the ganglia. Let me remind you, what does it mean? Here is like cross section of the spinal cord, and you know we have the you see here the roots, anterior root and posterior root, the nerve. So here is like an enlargement posteriorly for what we call it dorsal root ganglion, dorsal root ganglion. So this is a ganglion, right? And also this is a spinal ganglion, which is close to the spinal cord, right? We'll talk about it. And we have also autonomic ganglion. For example, when you say autonomic, that means you have sympathetic and parasympathetic, and you know on each side of the vertebral column, on each side there is like a piece-like structure in which you see here this another ganglion, we call it sympathetic ganglion, and you have many on each side of the vertebral column. We call them sympathetic ganglion, and we have also parasympathetic ganglion. So so if you look at this cross section here in this figure, guys, this is a nerve ganglion. It's a spinal ganglion, and you see there are like cells here and there, and they are surrounded by a thick capsule, which is a connective tissue, right? So as I mentioned, there are two types of ganglia. You have like this one, which is the spinal ganglia, and you have the autonomic ganglion. As I mentioned, we have here the sympathetic, for example, and you have the parasympathetic. Let us start with the spinal ganglion. If you take a cross section through this ganglion, guys, here you will see this is a spinal ganglion, and it's, as I mentioned, beside the spinal cord. This is the spinal cord, so it's okay beside the spinal cord. And look at it here. Here is like, for example, you get sensation from your skin, right? So then the signals or impulses will be transmitted through the nerves, then passes through the dorsal root ganglion there to the dorsal horn in the spinal cord. That means the dorsal root ganglion, if you remember at the beginning of this lecture, we said that the type of neurons located in the dorsal root ganglion, we call them unipolar neurons. Unipolar neurons. You have a cell body, then it has one process, then the one process will divide into peripheral and central processes. Okay, so that means these spinal ganglia works or function as a relay for different sensation before entering into the spinal cord, right? For example, the sensation from your skin to the spinal cord. Okay, now look at the nerve cells there, or what we call it because we talk about the spinal ganglion. That means the type of cells here or type of neurons are unipolar neurons. This is number one. And look at the body of the cell with the nucleus here. They are surrounded by satellite cell. At the beginning of the lecture, we mentioned that there are glia cells in the peripheral nervous system, Schwann cells and satellite cells, and we said that the satellite cells, look at them, they are extensive there, and they are surrounding the body of the unipolar neuron to nourish and support it, although still it's not really understood what the main function of the satellite cell there, but mainly they support and provide like nutrition to the nerve cell. This is number two. So look at the spinal ganglion. Look at the here at the capsule. This is a connective tissue capsule, which is indeed in the spinal ganglion. It's thick, right? It is thick compared, of course, to the autonomic, which is thinner, right? So at the spinal ganglion, guys, we have unipolar neurons. The unipolar neurons like surrounded completely by the satellite cells. You see, these are the satellite cells, make like a ring, and they are unipolar neuron, and they are aggregated into groups. As you see here, they are few in number. That means the unipolar neurons in the spinal ganglion, the number of the cells are few. This is number one, and they are aggregated either in rows or like groups. As you see, they are surrounded by satellite cells extensively, and the what do we call it? The capsule is thick, right? So here's another like show. Look here, they are like in rows. They are like or in groups. Here is like in groups, and they are few in number, right? They are few in number. Which type of cells in the spinal ganglion? They are unipolar neurons, unipolar nerve cells surrounded by satellite cells. As you see here, look at the satellite cells, right? Okay. Now, sympathetic ganglion. Let us move to the autonomic ganglion. And let us talk about, because similar to each other, let's just talk about the sympathetic ganglion, present like a chain, as I show you, parallel to the vertebral column, or they can be in different region of your body close to that organ. Now, if you remember, we said that the spinal ganglia work as a relay station for sensation, but indeed the sympathetic ganglia, they work as a relay for different motor function. That means from the brain spinal cord you get the order, then it passes all the way to reach the sympathetic ganglion that means the order is a motor function, right? Uh, that means it's a relay for different motor function arising from a spinal cord in order to be distributed to the autonomic distributed the autonomic order to the different organ body organs, right? That means, for example, let's say a spinal cord here, and then the order comes from there, passes through the sympathetic chain, sympathetic ganglion. From there will be moved, for example, to the heart or to the lungs or to the vessels here or there, whatever, right? Okay, so now this is number one about the function and where it's located. And what about the type of neurons there? The type of neurons, it is multipolar, multipolar stellate shaped nerve cells. It's like a star shape, right? It's like a star shape. It's multipolar cells. Spinal While the spinal ganglion, the fibers are myelinated, right? But here in sympathetic ganglion, usually the fibers are non-myelinated. Usually are non-myelinated, and the shape of the cell is like the shape of the cell is multipolar, uh, stellate shape. This is number two. Number three, they are many and they are scattered. They are not grouped or in rows. No, they are scattered because they are many. They are crowded there. And yes, they are surrounded by stellate cells, but not extensively, not as much as in spinal ganglion. So just a few stellate cells there. This is number three. Also, look at the capsule. The capsule is thin compared to spinal ganglion. You know that the capsule in spinal ganglion is thick, but in the sympathetic ganglion, the capsule is thin, right? So the capsule is thin. You have here in the sympathetic ganglion many neurons. They are many, not like spinal, and they are multipolar. They are mainly non-myelinated. They are surrounded by few stellate, not that much as spinal ganglion. And what else? I think also here, look, they are scattered, they are not arranged, they are multipolar, star in shape, non-myelinated. The capsule is thin, right? And the size like small, the size of the cells like are small, but in the spinal ganglion they are like range from small to large. It's like it's vary. Here's like if you would like to read it, I don't want to iterate it again and again. This is a comparison between the spinal ganglion and sympathetic ganglion. As I mentioned, the location, the spinal ganglion in the dorsal root ganglion, but here is like mainly in the sympathetic chain, right? Here is like the spinal ganglia is like unipolar cell, while in the sympathetic they are multipolar. And in the spinal ganglion, the neurons like arranged in groups or in rows, while the sympathetic, no, they are scattered, not organized. In the spinal, mainly they are myelinated, while in sympathetic, as I mentioned, they are mainly non-myelinated. In the spinal, they are surrounded in shape, while in sympathetic, they are irregular because they are multipolar, while in the spinal ganglion, usually they are unipolar. In the spinal ganglion, guys, you have more satellite cells, but here's like less number of satellite cells. Also, one important thing here, guys, that the spinal ganglion, there, because they are few, they are away from each other, and there is no synapses between neurons. While in the sympathetic ganglion, there are many crowded, scattered there, so there are synapses. There are synapses between multiple neurons there. That means when there are many synapses, it means you need more blood. That means it's rich in the blood supply, but in the spinal, unfortunately, it's poor, right? Well, finally, I would like to talk briefly, although we published a paper recently about the blood-brain barrier. I will try to leave the link there. But let me give you briefly. We talked at the beginning of the lecture about it, but this is just a pale vessel that, guys, a vessel from the brain that shows is the RBC there. And look at the wall of the blood vessels in the brain, which is specialized, in which it's formed like lined by endothelial cells here, and the endothelial cells like connected by tie junction, right? Also supported by pericytes and the astrocytes here, and should be a neurons here. So all of these cellular elements form for what we call it blood-brain barrier. That means a barrier between the blood and the brain here. So this barrier, these cellular elements, including endothelial cell, tight junction, pericytes, astrocytes, neurons, all of these cellular elements form the blood-brain barrier. So the blood-brain barrier, it is anatomical and physiological selective protective barrier that regulates the passage of materials, ions, solutions from the blood to the brain and vice versa, because the brain is very sensitive and the concentrations of different ions and stuff there in the blood is not suitable to be passed, or it's not suitable to the environment of the brain or the brain tissue. So that means the concentration and ions should be regulated to pass with specific concentrations. Thank you so much. Hope you find value in it. Thank you.